Okay, so the last part of this lecture series is just kind of going to be expanding on the concept of endocytosis and exocytosis. Now, if you watch the video, the narrator kind of explains some of this, but let's go ahead and let's do this. So we've got endocytosis, and there are some processes that we need to understand. Of course, we define this as taking into the cell, but there are different types. So endocytosis, of course, is going to be receptor-mediated. Okay, so receptor-mediated. We talked about that one, and you even saw a video on that one. Okay, so there's going to be another one, and it's called pinocytosis, and this is taking in liquids. Taking in liquids. Okay, so that's kind of interesting. And then, of course, the last one, and she addressed this one in the video, but is called phagocytosis, and it is specifically addressing solids. So taking in solids. Okay, so you're probably asking me, well, what makes phagocytosis and receptor-mediated endocytosis different? And this is, I'm going to call this, and this is my wording here, but this is called a chemical match. So whatever the cell takes in has to chemically match the receptor that's on the surface of the membrane. So it's really, again, it's really specific, and not letting just anything in. I mean, if you don't have the proper credentials, you're not coming in. Pinocytosis, of course, I'm going to take in certain liquids, and of course, phagocytosis is taking in solids. And we see that in white blood cells. They'll engulf anything, and they have to, in order to keep it safe. And you've got to see what a lysosome does. Now, exocytosis, not a whole lot to really discuss there, except in the animation, you see the vesicle fusing with the membrane. Okay, so the vesicle fuses with the membrane, and it keeps things from escaping. It keeps things from running rampant. It keeps things from moving around. It keeps everything kind of contained. And we saw the vesicle when we were watching or looking at the Golgi apparatus. So if the Golgi apparatus is packaging waste material or packaging proteins or lipids that have been created by the rough endoplasmic reticulum or the smooth endoplasmic reticulum, it's going to do so using a vesicle. So this is kind of like a vehicle for transporting goods. Again, it's going to use energy to do this because these things get really specific. Now, the only time that exocytosis and endocytosis may not be using energy would be on the passive transport side, and that's when they're following the concentration gradient. So just keep that in mind when you're trying to decide. The next discussion that we're going to have is going to be based on diffusion. Okay, now I know you're kind of giggling because we've been talking about diffusion, but diffusion and cell size. Have you ever wondered why our cells are really small and why some cells are big, but not all cells are big? So we're going to kind of take a look at diffusion and cell size.